I invite the audience to go and stand. Thank you. Please, Francesca. Waarneemt. You may be seated.
So welcome everybody to this PhD defense, also the public here, and of course the candidate. And hereby officially I open this academic ceremony in which Ms. Francesca Perin will defend her academic thesis entitled Versatile Multi-Component Bioink Platform for Advanced Biofabrication. May I invite you to present a summary of your study and the conclusion of your thesis in about 15 minutes. And I'd like to give you the word.
Thank you. Thank you everyone for being here. I will give a brief presentation and guide you through my thesis. So let's start with a little bit of definitions of what is bioprinting and what are bioinks. Bioprinting used to be defined as 3D printing of organs and tissues. And as of spring this year, we finally have an official definition from ASTM that states that bioprinting is three-dimensional printing of materials called bioinks to fabricate constructs to use in biological or medical applications. So bioinks are some special materials that are composed always of cells and optionally, but very often, of biomaterials. They are processed by bioprinters that are nothing more than a 3D printer that is suitable to process these materials in conditions that keep sterility and the cells alive. And then we end up with a bioprinted construct. Here in the picture, what you see is an actual 3D printed living ear. It's a construct called Aureo by Print Bio, and I chose it not by case, but because I think it represents the state of the art of bioprinting in the clinic right now, because this is one of the few constructs that is currently under clinical investigation in the USA. So how is actually bioprinting doing in the clinic? In chapter 2 of the thesis, we went through the role of bioprinting in the regulatory landscape and how present it is in the clinic. And we found that bioprinting is doing very good in terms of research trends because the number of publications has been increasing in the past years consistently. And this is also reflected in a growing market. Here you see the number of patents in the last decades. However, this market of new companies making bioprinters and bioinks is mainly addressed to researchers rather than the clinic, because when we look at clinical applications, we still don't really have any that are really in the clinic. And this is, of course, because it is a novel technique and it will take time for it to get approved. But we also found that there are some points in which we could do better as researchers in terms of standardization and harmonization of the techniques that we help with translation. So then moving on with the thesis, we decided to look at the creation of bioinks with an eye on translation for the future. And in chapter 3, we decided to start with trying to standardize a way in which we select printing parameters for extrusion bioprinting. In extrusion bioprinting, we have a cartridge in which, when a pressure is applied, we have a filament forming, and when we add movement of the print head, we can create a shape following the slicing file that we used. Engineering this kind of material, we need to take in mind the rheology and the final shape fidelity. The rheology, so the viscosity of the ink that we will work with, is strictly correlated with the geometrical accuracy that we can achieve, as you can see from this picture from literature. In the middle, we have the perfect engineered ink, in which we have extrusion of a filament, and we can print a grid perfectly following the lines. Whereas if the ink is flowing too much, we will have droplets and the ink will collapse. And if the ink is not flowing enough, it will be very hard to extrude, and then we will have a bumpy filament. And in both the extreme cases, we have a loss of geometrical accuracy. To give some examples from daily life, this is very similar to what pastry chefs do when they optimize the composition of the icing they pipe, or it's the same thing that whoever engineered toothpaste to flow after the tube with ease, but then stay in place instead of going everywhere, did when they engineered it. So we decided to work in building empirical models on both rheology and shape fidelity of a series of ink. And by applying constraints, we found a printability window. And with the models that we created, we optimized printing parameter selection and also the composition of our ink by applying a function with the desirable values that we wanted for the shape fidelity. Now that we had found a way on how to select the printing parameters, we needed to work on chemistry, so on how to consolidate our hydrogens. And we came up with the concept of creating versatile bioinks, so to have one bioink with multiple applications, because having translation in mind, we thought that in the future, if we have one single bioink that could be adapted to different biofabrication techniques and different applications, after initial translation for one application, it would be faster to have it approved also for different ones. We identified 3 pillars of what we wanted for our versatile bioinks. So one was minimal functionalization, and this was to make testing faster. And of course, for it to be versatile, we really needed the rheology to be tunable, both before and after deposition, so that we could adapt it to different techniques, but also we could adapt the viscoelastic properties to different tissues after cross-linking. And of course, we would need to tune the biological response of the gel. So we tried to build versatile inks with 2 different strategies. In chapter 4 and chapter 5, we presented strategy 1 that I like to schematize with a Swiss Army knife, because that is one tool with multiple functions. And the ink that we selected are one tool. It's alginate tyramine. And we did so because this polymer is a multi-cross-linking platform. I schematized here on the right. Alginate tyramine can be cross-linked in different ways, both ionic and covalent, and via different reactions that give it quite a big adaptability in terms of biofabrication, because they have different triggers and different kinetics. So we did play around a little bit with the cross-linking and also managed to merge the best of both worlds of the enzymatic and the photo-cross-linking into a cross-linking that we called photoenzymatic, and that could be printed with extrusion bioprinting. And then to demonstrate that this was adaptable to a different technique, in chapter 5, we decided to print it with microfluidic bioprinting. Microfluidic bioprinting is very different than extrusion bioprinting. We have, here you can see a microfluidic print head where the inks flow. And for this kind of ink, it's very important that when they meet in that Y-structure junction, there is almost immediate cross-linking so that a fiber is formed and extruded. Otherwise, we just have droplets. And we could do this with the enzymatic cross-linking, and we were able to print. And I think this is a good statement of the adaptability, because the rheological requirements for this kind of printing are completely different than the ones for extrusion bioprinting that we presented in chapter 3. Now that we had an ink and we could form it, we had to work on the biology, because alginate, as we know, it's bioinert. So we need to give it something that the cells will like. And we did this in chapter 5, and we envisioned to do it in 2 different ways. The first one is for any molecule containing amines. We can graft them the same way that we grafted tyramine to the alginate, so during synthesis. And then any molecule containing a phenol can be added during enzymatic or photocross-linking via deuterosin bonding. We selected 2 molecules, 2 model molecules for this approach. For the amines, we selected the ferroxamine because it's a very well-known and studied hypoxia mimicker. And for phenols, we selected a commercially available RGD with a pending phenol molecule. We found that the RGD was actually successfully introduced, and the cells were forming focal adhesions. And we also found that the ferroxamine preserved its chelation properties. But unfortunately, we also found some issues. So the cells were not able to properly colonize the hydrogens because they couldn't really remodel it. And also the ferroxamine really heavily interferes with the HRP cross-linking. So when figuring out how to move forward with the issues that we found, we thought to move to a different system, which is presented in chapter 6, and which here I schematized with Lego blocks, because we moved forward to click chemistry, in particular to thiolene chemistry. In thiolink chemistry, we synthesize arginate norbornene, and then to create a network, we use dithiol crosslinkers. Here you can see pegdithiol, but we also use different ones that are described in the chapter. And this system had a very much higher tunability given from the modularity. So by changing the dithiol crosslinker properties, we could change the final properties of the hydrogel. And it was also very easy to use different peptides for achieving the biological functionalization. We designed a sort of matrix of experiments to check for the biological performance of the hydrogels when using different kinds of crosslinkers, different kinds of peptides, and then we studied both encapsulated cells or cells seated on top. And then here you can see a brief example of how different cell lines behaved very differently on gels when we changed the composition. So here we have UVEX, HMSCs, and Arpitex on gels that could not be modeled, could be modeled but didn't have RGD, and had RGD and could be modeled. So these experiments served us to select the optimal conditions that we needed, and then we could move forward for bioprinting. And for bioprinting, because all this work was carried under the Birdie project that aims at mimicking in vitro the kidney, in particular the nephron, we were aiming at printing capillaries and tubular structures. And we selected once again to use the microfluidic bioprinter, but this time with a more complex printhead that allowed us to produce hollow fibers by use of a sacrificial core. And the next step forward that we did is that due to preliminary experiments from the Birdie project showing the cells suffering a little bit when printing with alginate and ions, we decided to try and print just with photocross linking. And this was achieved and was optimized by a design of experiment approach in which we studied how the different printing parameters had an effect on the outer diameter and the thickness of the fibers that we could produce. Here you can see two examples of fibers that were made fluorescent, and you can see that they have more or less the same outer diameter, but they have a very different lumen. So after achieving more or less control on the size of the fibers that we could print with these cell-free experiments, we moved on to printing with cells. And here is an example of UVEX printed after 2 weeks. What you see here is the surface of one tubule in which the UVEX formed a sort of monolayer. And you see that we had a very good distribution of CD31 and bicadirin, which is a good sign of tight junction formation from endothelial cells. So to conclude the overview, this thesis was aimed at finding strategies to try to help translation in the future. And in chapter 3, we found that empirical models were very useful tools to optimize printing without relying on trial and error. Then we tried to build versatile bioinks, and in chapter 4 and 5, we found that alginate tyramine is easily adapted to different bioprinting techniques. However, it still needs a little bit of work on the biological side. And finally, in chapter 6, we found that alginate norbornene was very highly tunable, both in terms of cross-linking kinetics and viscoelastic properties, but also on the biological performance. I would like to thank all my supervisors, and because I'm enrolled in a double degree, both my research groups, so Biotech from Trento and Carlos Group, CTR and Merlin from Maastricht. And then there are some special thanks to people that contributed to the chapters. And thank you.
Thank you very much, Ms. Candidate, for your well-illustrated and clear presentation. It's nice to see. I also would like to mention, indeed, like you mentioned before, that this is a double degree between Maastricht University and the University of Trento, and we are always pleased, especially with according to new EU laws, that we're going to stimulate double degrees in the EU. We're going to, before we start the opposition, I'm going to introduce also the promotion team, who is sitting opposite of me for the audience. And there are 4 members, of course, 2 from Maastricht, 2 from Trento. That's the way it works. And I'd like to introduce, and I start here on my left side, for you maybe on the lower side. Professor Maniglio. He is professor in functional materials, tissue engineering and surface modification from the University of Trento. We have Professor Motta, who is affiliated or professor in biomaterials and again tissue engineering and fabrication, also at the University of Trento. We have Professor Motta, and he is affiliated with the Department of Additive Manufacturing and Bioprinting for Regenerative Medicine. That's quite a lot of wording. Thank you very much. And of course, the first promoter from Maastricht is Professor Moroni, and he's professor in biofabrication for regenerative medicine. And all four have worked with the candidate over the last years to bring to the result of today. That's basically the question. For the audience, we have 6 members in the corona, and one of the members is online, is in the screen behind you. But if he's going to ask questions, it will be also seen on the screen in front of you. And I would like to start with the first member, who was also of the corona, who is also the chair of the assessment committee, which is Professor van Grinsven. Professor van Grinsven from Maastricht University here is professor in regenerative medicine, and I would like to give him the first opportunity to discuss with the candidate. Professor van Grinsven.
Thank you. Dear candidate, thank you for this excellent presentation and for the very nice thesis. I read it with great pleasure. I'm not a biomaterial specialist, but I learned a lot from that, and I would also like to thank and to congratulate the supervisory team for that. As I'm not a biomaterial specialist, I'm going to chapter 6 with you. When we look at your experiments with the RGD peptide, I was quite intrigued by it, because you see that the endothelial cells very nicely adhere to it, make nice structures, and when you look at the rhinoepithelial cells, they absolutely didn't like it. They made like little aggregates, and you could really feel that they hate it. So I found it very strange, because normally the cells have integrins and they would really sit on the RGD. So do you have any explanation for this behavior?
Highly esteemed member of the committee, thank you for your kind words and for the question. I indeed also was surprised the first couple of experiments that this happened. And at the beginning I believed that something was going wrong. So you see some of experiments presented here, but there are so many that didn't make it to the thesis because I was not understanding what was going on. But at the end, after doing I'm not a biologist, but from literature research, I found that kidney epithelial cells do present some sort of weird behavior connected to the presence of integrins and soft hydrogels, in which they form these cyst-like structures instead of forming a monolayer. I'm not exactly sure why. There also, I found some papers, one from the group of Kaplan, for example, in which they tried to demonstrate that an overproduction of RGD was connected with the cystic formation in kidneys. But there is not so much yet on that specific why it happens. However, there are some groups that are using this to produce tubuloids instead of using common organoids. So cultured in soft hydrogels such as matrices and in presence of RGD, the cells tend to form aggregate into these tubular structures that are very good to culture primary cells and patient-derived cells in the long term. So I don't think that is necessarily a negative sign for the biomaterials that they did form cysts. However, indeed it is a negative for printing them in tubules. I did find recently a paper in which someone used this to make them self-assemble into tubules. So they did print a sort of sandwich of very soft biomaterials and just printed a very dense line of cells in the middle, and they self-distributed into a small tubule, which I think was a very nice way. If I found it earlier, maybe I would have tried something similar.
Because it actually, when it comes to one of my next questions, so when I looked at the bioprinting with the endothelial cells, they were on the inside and on the outside, yeah, which physiologically you don't want. So how do you want to combine it, and especially when your epithelial cells make these cysts, that would not really fit to that?
Yeah, I think indeed they did a little bit less cysts when we tried to change peptide. And also I feel like when we culture them in the tubules rather than in the gels, maybe because we kept them for a longer time. They did form also a sort of monolayer. I think to move this work forward, we really do need to work on exactly why this is happening and how to prevent it. One way could be also to not try to prevent it, but try to use it, as they did in the paper that I mentioned. So for example, you could give them a very soft RGD hydrogel in the core that they can remodel and that you can remove at the end, so that they make the tubule by themselves, I guess. And regarding the endothelial cells, I do think that we have to prevent them from going and doing the double layer,
Right.
because that's not what we wanted. I think that could be prevented in the future, maybe by using a more complex print head. So you can do 2 layers. So you have the sacrificial core, one thin layer with endothelial cells, and then 1 layer that they cannot remodel. So you force them to stay in that tube.
And the tube you made, do you think there's flow through? Because in some of the pictures, you have some tube, some cells, some tube. So do you think it's really functional?
That's a question we really didn't answer yet. I know that people left in Carlos' group are working on this right now, on trying to perfuse them, because I also think, going a little bit back to the previous question, that perfusion could help to have also the preferential growth just in the inner lumen, because you keep giving them fresh medium. But I'm pretty sure that the ones without cells were hollow all the way through. We do have to check what happens when you culture them for 28 days. It's a long time, and they do remodel the ink, so it would be nice to check what happens indeed.
Last short question. What do you actually want to achieve? Do you want to make a complete kidney, or what is the...
Oh, no.
project you want to make?
Good question. No, we just want to mimic the proximal tubuli, so it would be 2 channels, one with the epithelial cells, one with endothelial, in close proximity, in which you can flush blood, urine, and check for the exchange.
Thank you very much. I'm happy with the questions, with the answers. Sorry. I'll get back to the program.
Thank you very much. Then the opposition will be continued by Professor Le Visage. She was a member of your assessment committee, and she is professor in regenerative medicine and skeleton at the University of Nantes, or Nantes Université, I should pronounce it correctly. And I would like to thank her already that she joins us today, and I'd like to give you the word.
Thank you. First, I would like to congratulate the candidate on her excellent presentation and thesis, and to thank, of course, the whole team for the invitation to be part of this committee. I have several questions. I think the first one will be actually on the chapter 3 regarding the model that you developed and the printability. You mentioned that you are developing this model for a monolayer structure. Do you think it could be actually applied to a multi-layer structure? And did you take into account the fact that there is a risk of delamination whenever you are printing a multi-layer structure?
Highly esteemed member of the committee, thank you for your kind words. We did indeed develop it in 2D, and kind of on purpose, because we wanted to make it a tool that is kind of easy and fast to use. And then, of course, it's not perfect to translate it into 3D, but we tried. So we developed it in 2D to have nice accuracy for a while, because it takes time to take the pictures and so on. So we envisioned that if we developed a way to cross-link layer by layer within a few minutes, then it would be fine to go into 3D. But of course, and we tried in the paper, there are some 10, 20 layers structures, I believe. It's true that this is not a modeling of what happens in 3D, so there are risks on using this approach. So yeah, we could then further enrich the model going 3D and adding also other tests. For example, so in literature, a lot of people use the pillar test, in which you print on pillars that are farther away from each other and check how far you can go. It would need some implementing, of course, and to add it.
My second question will be based actually on the development of novel biomaterials. I've seen in your thesis that you mentioned that there are on the market several human-based products based on the decellularized products, usually from adipose tissue. And I have to say that it seems to me that these products are not well known and not very used in the research in our field. Do you know why? Do you think that it's just because these products do not have any future and that it's better to develop synthetic polymers, or do you think they have a future?
Yes, I was also very intrigued by this, also because all the companies that I found that are cited in the thesis are in the US. So I was a little bit intrigued by why is it in the US and not here. And unfortunately, I did not find like a proper peer review material about this. I only found like stuff from FDA and so on. So apparently in the European Union, both for regulatory point of views, but also probably culturally, people are less prone in wanting to use human-based materials. I also personally found it a little bit weird that it's possible to commercialize human-based materials, honestly. Of course, they come from tested banks and so on. They're not shady, let's say. But the other thing is that in the EU, because of how our healthcare is built, the clinicians are less prone to use very expensive materials for procedures, whereas in the US, where private medicine is more diffused, there is a bigger market for materials that might be very expensive. So from my understanding of what I found in between the EMA and so on, is that so far their superior potential in healing has not been demonstrated enough to justify their bigger, expensive, and possible ethical remarks about it.
So that's a really good rationale for developing novel polymer.
Yes.
That could be very, very useful. Talking about the social impact of your of bioprinting in general, you started your presentation showing that here, and that was actually one of my questions based on the fact that the clinical trial that have been approved, most of them, the recent one that you cited was approved for compassionate cases. And I'm wondering if you think that there is a chance that bioprinting product could be extended to a more general public or patients, or do you think it will be always restricted to this type of really, really compassionate cases?
So I think eventually it will not be just for compassionate cases, because I think it's stated also in chapter 2 that I see that the regulatory bodies are really working around trying to make it easier. In general for ATMPs, not just like tissue engineered products, not even just bioprinted ones. I do think it will take time. Also, yeah, in the Aurinovo or Aureo, they changed the name now, but the year. I think they did incredible work in finding a way to work around some issues to get it to the clinic. And I think because it's an easy organ, so I'm not saying it's only aesthetic, but mainly aesthetic, I would say. I see how they could easily convert it if it gets approved for other applications like nose or so I think that's it. But probably it will take a long time, because the clinical trial for Aurinovo was 11 children. That's not a very big group. The clinical trial will finish around now, the first phase, then there is phase 2. No one knows how it went so far because it's still ongoing, so I think it might take a long time, and I'm not sure we will see anytime soon. Often there are these images of bioprinting with like a printer printing a heart or a brain. I don't think we will see it anytime soon. That's too complex for the moment.
And my last question will be actually on something that you mentioned in your thesis regarding the recent draft that was updated from the ASTM committee regarding the assessment, the test that should be performed on bioprinted construct. Can you tell us a little more about this update, and what do you think that these tests are going to be very useful for the translational point of view of this product?
You mean the one that I said that?
Yes.
Yeah. I think that standard right now, in terms of like practical terms of I'm in the lab and I need to test something, is not very useful yet. It might be useful for someone that is completely new to the field, because they basically collected practices that we already did in the lab from literature. But I think it's a good sign that they know that this is in there and we're working on it. It also took years to do it, because when I started my PhD in 2019, that was in draft. So it took 5 years to publish it. I hope this is a good way for them to come faster and to finally have like proper standards like you do for other materials. For example, if I'm 3D printing a thermoplastic polymer, I will find very precise standards on how the dog bone should be, how I should tensile test it, and so on. And I hope that in a not so far future there will be something like this also for hydrogels and for bioprinted hydrogels, in which everyone does the same test, and then the results that you get are very easy to compare. And I think that will help a lot to move faster.
Well, thank you very much for all your replies, for the quality of your replies. Really appreciate that. I'm done with all my questions. I will give you my point back.
Thank you very much. Then the opposition will be continued by Dr. Tirella. She is affiliated with the Department of Biomaterials and 3D Printing Technology at the University of Toronto. And I'd like to give you also the word to ask some questions to the candidate.
Thank you, and thanks again to the candidate for a very nice presentation and comprehensive work. It is a really hard job. I have only a few questions, more based on the material part, as you know my background. In your research, you used a lot of modifications of alginate, as it is well known as a really good candidate for bioprinting, but has a lot of drawback for working with cells. And in specific, in chapter 4 and 5, you used different techniques to characterize the bioink formulations. For example, the degree of substitution or the linking of DFO. Just my question is on how do you think these like FTIR and MR spectra that you use are sufficient to corroborate hypotheses, hence predict the quality of your printing during both the extrusion-based and the microfluidic 3D printing technologies?
Okay, thank you, esteemed member of the committee. Good question. I think when you use chemistries like tyramine or, for example, metacrylation, the degree of substitution might have a huge impact on the final modulus. And I don't think it's always that easy to predict, especially when you're printing, for example. You can do very nice photorological measurements by changing the time you irradiate, but then how does it reflect on how long is my filament being irradiated? If I'm, for example, exposing it layer by layer, how many times do I actually expose the first layer to some UV compared to the last layer, and so on? I do think that because of this, the update to click chemistry was nice to solve the issue, because with norbornene, and we tested it, I did put enough, like a big excess of the amount of norbornene that I would need to mimic the modulus of kidney, so that I always had a lot of free norbornene for 2 reasons. So to be sure that I was grafting all the RGD that I wanted to put in there. But also this way, the oscillation of the modulus drops a lot. If you control the amount of cross-linker that you introduce, then when the reaction goes to plateau and it goes very fast, you can irradiate it longer or less, and it's going to be the same. So I think for some kind of inks, it's very hard to predict exactly. Like for tyramine or metacrylation, a 2% difference might result in a slightly different modulus. If you work with click chemistry and leave some groups unsaturated on purpose, you will have a much higher precision on the modulus.
Okay, thanks for that. I think this really linked to my next question. That was on the coupling of thiamine and the 2-step carboamide coupling, and on the pending of RGD that are added to the hydrogels during that cross-linking. Do you think the concentration you've used of the RGDs, then peptide, is sufficient to trigger then biological processes further down the line? And how did you characterize, or can you characterize precisely this amount in the hydrogel you have obtained?
Thank you for the question. I did not quantify how much RGD I used, not in chapter 5 and neither in chapter 6. To be honest, I just, indeed, that's something that should be done further down the line. I did use amounts of RGD, like I worked with concentrations that I found in literature, which they saw results, and I saw that there is more or less a consistency on using the around 100 millimolar of RGD. So that's where I started from. I tried a little bit less, I tried a little bit more. I did characterize a little bit of the amount of RGD now that I think about it, sorry, in chapter 6 with the fluorescent tag. Yeah. So technically I could have done it also with the Tyramine if I bought a fluorescently tagged RGD to graft in there. But at that point, yeah, I already had moved forward. I still think that the biggest problem in there was the impossibility of remodeling, because they did form little arms going to the RGD. They just didn't do anything afterwards. But I do think that going forward with that ink, we should characterize this better.
And do you think this would help then to understand all the biological tests you've done further?
Probably, yes. Yes.
Do you observe any variability between samples in this case when you were attempting to add
the RGD? Yes, but there was some consistency of, for example, never seeing the cells changing their shape if there wasn't RGD, and they doing something from 100 millimolar onwards. That was very consistent in between experiments. I saw some weird behaviors, I would say, or unexpected behaviors with the metabolic activity or the Alamar blue that I still am not sure if it's because I saw it consistently that the more RGD, the lower metabolic activity, and that really confused me, to be honest. But it happened for several experiments, so this is something that I would like to further investigate, given the chance to know what it is.
Then that's my very last question, and given the complexity of the whole thesis, so moving from biomaterial ink design to different cross-linking techniques you've used, bioprinting process, biological evaluation, etc. What I want to ask you is if you can elaborate a bit more on what could be useful to add on the characterization of all this holistic approach on the 3D printing process, and if you think that there's any possibility that you can inform with control system approaches or neural network or similar strategies to help in harmonizing the overall process that you described, and in particular if you think that these would help on the complex design of the microfluidic bioprinting head.
Okay, yes, so starting from the holistic approach, I think indeed machine learning and so on will have a role in defining how we do this, and I do think that harmonizing testing procedures and probably also maybe building in the printers some softwares to kind of have a software that is doing it and not the operator would help. I don't remember the name of the authors, I'm sorry, I think I cited it in the discussion. There was a very nice work that I think kind of covered this from Germany. 13 different groups doing bioprinting teamed up, and one single group produced the inks, so they were all the same, all from the same batch. They did the SOPs and they sent them around, and there were 13 different groups printing following the same SOP, and they also developed, based on machine learning, a procedure to evaluate the quality from images of the hydrogens that were printed. They also, of course, gave the G codes, and they still found variability of what they printed. And I found this very nice because they also found some things that were causing variability. For example, the fact that some printers have manual calibration, so you have to decide how far you are from the plate, and that was a source of variability. And they really advocated also for having a higher standardization and harmonization on how these preliminary steps are done by the printers themselves, which I think was a very genius idea. So indeed, if we could buy, for example, an extrusion bioprinter with a little bit built-in camera in which you put your ink, and the printer tries itself different pressures, different speeds, analyze the images, and tell you, you print this with these parameters, that would be very nice. I see it a little bit harder with different printing techniques for the moment, but why not?
Okay. Thanks again for your opinion. I'm really happy and pleased by your answers, and I'm happy to give back the...
Thank you very much. Then we continue swiftly to Dr. Gans Fari. She is Associate Professor of Biohybrids and Medical Textiles at the Aachen Maastricht Institute for Bio-Based Material, and I give you the opportunity also to have some questions to the candidate.
Thank you, Mr. Chair. First of all, congratulations, dear candidate, for your excellent presentation and also your beautiful thesis. I really enjoyed reading it, and also congratulations to the supervisory team for such a nice work. To follow up on the previous question, I would like to ask a general question about indeed the first main chapter of your thesis related to the regulatory framework of bioprinting. I would like to mainly focus on the natural or a natural-based hydrogel. That's also something that you used in your thesis, and of course we know that these hydrogels are highly valuable in bioprinting due to their intrinsic characteristics. But we also know that there is limitations in terms of because of their variability, especially concerning the batch-to-batch consistency of these kind of hydrogels, which can significantly impact their properties and finally the performance of the final product. Since you use these hydrogels in your thesis as well, I was wondering what strategies the field of bioprinting basically can employ to maybe not overcome, but like to minimize these challenges. And if you, well, of course, you also mentioned the AI tools, and I was wondering if you can think of like techniques or tools such as AI tools that can help us address these limitations.
Esteemed member of the committee, thank you. So indeed, variability is an issue, especially with natural-based polymers. I would say with some of them more than others. For example, I did use alginate, and of course I might have variability on the molecular weight and so on, but something that is nice of using a bio-inert polymer is that I will not have variability on the biological cues, because I'm the one that is putting them in there and deciding how many they are and so on. I would say that a strategy to try, because we cannot completely avoid it, I suppose, so a strategy to minimize it would be, I think, to try and have a lot of control when characterizing on the groups that are the ones that are more important for your final application. So for example, as I mentioned before, what I liked about the alginate norbornene was that, you know, I just put more norbornene in there, and then I had a very high control on the modulus. So using a different material, I would try to really think and target which are the groups that, when they vary, will affect my final outcome, I guess. Although it's not probably a perfect solution, it could help. Then I guess, but I don't know if this should be done by the field of bioprinting, but in general, I think that there is also a lot of work going on trying to synthesize this kind of materials and in a different way that could help. For example, instead of extracting alginate from algae, having it produced from cultivation of microalgae, then you get a little bit more control on the molecular weight. I think it gets easier the more complex it is. For example, with protein materials, of course it's easier because their structure is much different. And indeed, because there are so many things to try to control, I think probably AI tools or machine learning could help with identifying which variability, like which groups varying will have the biggest effect on the outcome, and trying to minimize or trying to find strategies to cope with the variability somehow.
Okay, thank you. I should say that I was really glad to see such a comprehensive chapter on this topic because especially the discussion on like regulatory or translational challenges is always like lacking in academia, and I guess everyone who basically work in this area should be aware of these challenges. But what I was missing was a brief discussion on how, like, your studies in the subsequent chapters will address these challenges. And now I want to specifically ask you about chapter 4, where you used photoenzymatic cross-linking to basically have your opinion on what do you think would be the translational challenges if you want to bring that technology to the clinic, or if you want to translate it to the clinic, basically.
Thank you. So the chapter 4, it's a work that came out of realizing that photo cross-linking was super slow with the tyramine, so that it would take minutes to irradiate and cross-link. Whereas the enzymatic was very fast, but then sometimes it's hard when you have a burst reaction to actually print it. And that's why we merged it. In bringing the photo cross-linking to the clinic, it's kind of there already for different applications. So it's done for corneal cross-linking with riboflavin, exactly. So for, I think, keratoconus and other issues with the cornea, it is already FDA approved because it's safe for the eyes, also with a very long irradiation, and generally pretty safe for the tissues, I would say. So I think, for example, to bring that aspect in the clinic, we have something that is already approved, so probably it would be faster to try and demonstrate that this is something that is safe for the body. Also, vitamin B12, it's a vitamin, so it's easier, I guess, with some molecules to get them approved. The tyramine functionalized alginate would need intensive testing to show that it's safe, because alginate is approved in clinic for some things, but it's so far native alginate. From my knowledge, at least, there is currently no modified alginate in the clinic, also for different applications. So that would indeed require a lot of testing, and this goes for any modification that we use in general. It's just at some point, without modifying molecules, I think it's very hard to make them into a bioink, I guess. So I do think that it would take a long time to take something like that in the clinic. But using bits and pieces from things that are already in the clinic probably will help to put it together, I think.
Okay, thank you.
I'm sorry to interrupt.
Yeah.
We have to proceed.
Yeah, that's
I'm
fine.
sorry. Yes. And we have still two more to go.
Yeah.
So we continue now online to Dr. Kaspar. I'm not sure if he goes to the other screen as well, to the technical team. Dr. Kaspar is involved in the Department of Biomimetics, Biological and Living Materials of Aveiro University in Portugal. I would say welcome, and I give you the word.
Thank you so much, Mr. Chairman. Good afternoon, everyone. Good afternoon, dear candidate. I would like to say that I really enjoyed reading your thesis. It was an excellent work, and it was beautiful put it together with different chapters and the different synthesis of different polymers. And I would like to follow up on the previous questions and actually to ask you regarding your very comprehensive first chapter on the regulatory aspects, which are very well needed to be discussed within the field, within not only in biofabrication, but for example, in the whole tissue engineering and regenerative medicine field. And I see that you really focus nicely on the bioprinting aspects, but in your chapter 6, you use bioprinting as a technology to actually create an in vitro model that could be used, for example, as you have said before, to screen different types of molecules that are being mass transferred during your tubuli or to test different drugs. So I would like to hear your opinion on what would be the regulatory hurdles that you find on using bioprinting to print models that could then be used to translate different types of drugs into preclinical trials and then to clinical trials. I would really love to have your opinion, because I really feel that this is the edge of the discussion of the field.
Esteemed member of the committee, thank you for your kind words and your question. And indeed, I did not touch that much upon clinical testing, but just medical devices in the review chapter. You're right. I do think that probably building models will get approved, like clinical models that are bioprinting, will be approved before devices. Because of course it's less risky, because the main drawback in approving something in the clinic is for it not to be safe for the patient, obviously. And when it's a model and not something that needs to be in the patient, I think it's already a little bit easier. And also, I think because there is a lot of discussion in overcoming animal models and in the fact that the models that we currently use are not precisely reflecting what is actually happening during disease or in general in the body. I think this will also be pushed both by the FDA and the European Union into trying, I mean, they are already pushing into trying to let go some animal models and building something different. And in this, I think bioprinting is having and will have a big role. But again, the regulatory bodies need to come forward and establish rules on how you define if a model is suitable to substitute what is out there already or not. Because I think right now there is a lot of discussion, a lot of saying animal models are not reflecting what is happening, but then at the end, to commercialize something, you need to do animal testing. So having a very nice model that you cannot use for real life applications is not that useful. But no one is going to use them until they get somehow approved. So I think there is a lot of discussion, and I think it's moving in that direction, but I think we need some regulations for it to actually get there.
I agree with that, and thanks so much for your answer, because of course qualifying these models and seeing if they have real correlation with the in vivo scenario is also a challenge. So thanks so much for that, dear candidate. So I would like also to focus on your very nice chemistry during the thesis. I really enjoyed the different types of chemistries you used in the biopolymers, the way that you used photo cross-linking as a holistic way of generating these biofabricated constructs. And I would like to have your opinion on, you use alginate and then you use RGD to actually imprint some biological activity to your biopolymers, which are generally quite inert as per se. And I'd like to hear your opinion on the different strategies that are right now being used with tyrosine cross-linking and even norbornene to cross-link proteins into these biopolymers. So you can have, for example, DCMs cross-linked with alginates. I would like to hear your opinion on that. And if you have to choose one of those cross-links, which one would be if you wanted to add, for example, DCM as a whole biological bioink that is actually giving more signals to cells than just RGD?
So yeah, I decided kind of on purpose to use an inert polymer so that I could have a black canvas and I could select what I wanted to put in there. Also in view of making it versatile. So indeed, DSEM is much better to culture cells. They like it more. It's kind of their natural environment, but it's a little bit tissue specific. That's why I didn't use it. And indeed, there are strategies out there to cross-link DSEM, and one of them is actually to use the riboflavin and the tyrosine cross-linking. Also using photoinitiators. There is not just tyrosine, but also other groups that can cross-link. I think it's a nice approach. I think it's probably easier in this case to use something like the tyrosine cross-linking, or anyway using a photoinitiator and irradiate and see what binds, rather than click chemistry, because to use click or an approach like norbornene, even if you have some proteins that have the right groups, I think in the big complexity that is DSEM, it would be hard to know exactly what kind of groups you have, how exposed they are and available. So I think probably what people are doing now is just to cross-link and see what happens and do some experiments to check the final outcome. So indeed, for this, I would go for the tyrosine cross-linking rather than the click. It is less reproducible, though, I think, in general. Like, you can make it a little bit stiffer, but I'm still not sure that you can exactly control how stiff your DCM will be.
Yeah, I agree with that. Thanks so much. This is a very interesting discussion. I also think that the dityrosine is quite versatile because you don't have to change the proteins, but of course it's much more not reproducible in terms of the click chemistry, which is why orthogonal per se. And I would like, if I may, just to have another question about your chapter.
Due to time limits, and we still have a sixth opponent.
Thank you so much. And it was a pleasure to read your thesis. I'm just so sorry not being there, but it was a real pleasure.
Okay, thank you very much. Then we continue to Professor Horning. He's involved in our Department of Analytics in Systems Imaging at our university, and I'd like to give him the final moments for opposition.
Thank you, Mr. Pro-Rector. Dear candidate, first of all, congratulations. I think you covered a lot of different sciences. It's always dangerous, but I like people who take this challenge. So compliments for that, and also for the supervisory committee. I would like to ask one of your paranymphs to read proposition number 7.
You can move there. Or you can stand up and speak loud.
Yes, I'm doing very loud. Bioprinting research and market are running faster than regulators.
Yeah, so I have been reading that sentence 3 times, 4 times, maybe 10 times. My question is, so researchers are running faster than collaborators of the FDA in the marathon, or?
Highly esteemed member of the committee, thank you. I think I wanted to portray how I think right now FDA, ASTM and so on are always coming a little bit after with stuff like the ASTM that I showed before, that are collecting practices that researchers are already...
So regulations are always following the new technology.
Yes, they're taking a little bit too longer.
But then you agree that people in pharmaceutical industry are even running faster than bioprinter researchers?
I think people in pharmaceutical industries right now have not yet a very big interest in bioprinting itself, because
there's a lot of extrusion of polyethanol with tesogestrel, or we are speaking about naproxen, for example, hydrogels for children. It's there already for 20 years. So they do a lot of extrusion there. So they are doing this already. So
bioprinting for drug delivery rather than for culture.
Yes, of course. Yeah.
Oh yeah.
Yeah, but it comes, we are not speaking about the technology, but about regulation. So in terms of regulation and understanding of engineering, they are running faster, or still you disagree?
In terms of engineering, I think it depends. I think they go faster on what they can sell, of course.
I think they are faster, because now I have a second question on that. So I will push that you will agree with me, of course. Proposition number 1.
Harmonization is the key.
Harmonization is the key. I completely disagree. And actually you gave already the argument for that, because in Germany, and let's say that the Germans are always strict, they tried at 13 different labs to reproduce an harmonized soap waste method, and they were not even able to do so. So I would say harmonization is not key. Understanding of processing is key. And in process control, now I come back to my pharma, because in pharma it's called quality by design. So actually they do not test at the end, but already in the beginning and during. So now you agree with me, they are faster?
Maybe.
Maybe still?
I think, well, okay, yeah, you make a good point for pharma. Maybe I was focusing more when I also wrote the proposition on tissue engineering rather than pharma itself.
But then you can say harmonization specifically in the field of bio.
No, I still think harmonization is the key in general. Maybe it's because I'm an engineer, I'm not sure, but I think you need to give a framework on how you produce and you test things.
No, I completely agree. And for pharma, there are the ICH guidelines in order to take harmonization.
But you get approval to sell a medical device or also a molecule, whatever. For example, the GMP certification is not given by a set of rules. Like you have to build your lab like this.
Oh, exactly. So it's harmonization on guidelines.
Like they check it case by case, and every year they will renew your approval if you face it or not. So it is true that they do look into specificity of every different production place. Yeah. But still, they look into it by an harmonized set of rules of what is the outcome.
Normally, not only about safety, but especially about money.
Yeah.
And it also may be interesting to say that in many cases for for example, naproxen or gelatine-based hydrogels. Of course, people in the Western world take pig gelatine, but many people in the world will not use pig gelatine for naproxen distribution. And then suddenly your whole production process is not working, although you harmonized. So, no, last time? Let's skip that then. So, you believe in DOE, I absolutely believe you're right. So in that sense, I think chapter 3 and 4 are absolutely in line with that. But maybe also going back to the field where I'm in, it's analytical chemistry. In chapter 3 and 4, I thought, oh, I miss so much my analytical chemistry. And then I was very happy that in chapter 4 it was back. And then you showed me that you compared two batches, one with different structures. You used GPC, and you had two distributions of polymers around 100,000 to 1 million. And there was absolutely no difference. Then you look to the NMR, and of course infrared per definition by law is not showing any difference. So why didn't you use any mass spectrometry?
I think at the time it was not readily available for me to use. So I didn't go in that direction, and then again when
okay, so you have to ask Professor Moroni to have a mass spectrometer. Okay.
Hora asked.
I think you finalized your answer. I think yes, so we can proceed. Yes, good. Dear Miss Candidate, dear Miss Perin, the time appointed for defending your thesis has now passed. The degree committee will not withdraw, but will stay here.
Yes, please.
To discuss the quality of your thesis, but in particular the way you have defended your thesis in the last 45 minutes. I request that your company will leave the room here. Exactly. I will now make a little change here in this program, and you have to wait together with your audience outside the deliberations, and then you will return here back to the audience. Yes. Thank you.
Please, Francesca. Paranims, please. Audience, please.
Een beetje aanpassen. Die tekst is volledig
Audience, please be seated.
Very well. Welcome back. Dear Miss, I still call you Miss Candidate, dear Miss Perin, the degree committee here present has assessed the quality of the thesis, but in particular the way you have defended your thesis this afternoon. And we also look to the quality of your presentation here. And in view of its positive verdict, and taking into account your previous qualifications, the degree committee here has decided to grant you officially the degree of doctor. Professor Moroni is authorized to confer upon you this academic distinction in accordance to Dutch university customs, and therefore I invite your supervisor now to take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times?
I promise.
That's second before promising completely. To be careful and honest, transparent, independent, and responsible.
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Francesca Perin, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, fixed with the official seal of the university.
The photographer can proceed. Come here. It's important that you're there. Don't be shy.
In front, and maybe you can also put yourself.
Please be seated.
I'd like to give the words, 2 words, and we start with Professor Mota, followed by Professor Manicrio for the laudatio. Professor Mota.
Dear Francesca, dear Dr. Perin, what a hike. It seems that you went through several hikes throughout your PhD. You did the most hikes at ease in the Trentino mountains, but the last one was the easiest. As in Maastricht, the mountain, or better, the hill, is the St. Peter Hill, which is just 171 meters above the sea level. For this last small hike to Maastricht, you did quite several scientific hikes. The first one was the sulfation. That one was the Everest of this world. Unreachable, unreachable. We had to put that aside for the time being. With other reactions available in the institute, you placed yourself at work to quickly find an alternative. The alternative was alginate norbornene, which was a great platform to explore yet another unknown, the kidney. With the Birdie team and with a master student that you supervise, Anna, you did an incredible effort to make the best formulation possible for each cell. Another challenge was the bioprinting, but in this regard you were already experienced. The only thing that you didn't like was those 2 amazing LEDs that were at times not collaborating with the cross-linking. Together with Luca, another master student, that you explored yet another material, so that there is much more out there than the great work that you showed today in your thesis. The cell work was not your favorite, but together with your colleagues and students, you did an amazing job along the way, and you are now an expert. During your time in the Netherlands, it was clear that you improved your skills and perseverance and patience, for the technical to the emotional. You were always a great team member for the Birdie project, and along with Sveva, Sandra, and Alan, and many more, you left a legacy of materials for them to progress with their own research, which I'm sure will yield excellent results beyond the ones shown throughout your PhD and that you presented today. Your work was evaluated by world references, and achieving your doctorate is a logical sequence. The improvement on the diverse skills and skill sets that you have become expert with the interdisciplinary requirements known to the field were well mastered, and you demonstrate that today. I'm sure that your new research position at Polymath in San Sebastian in Spain will allow you to excel even further and give you the opportunity to climb those great mountains that science and research might bring ahead. I'm sure you will address any challenges with great responsibility and maturity. We wish you to continue to excel in the future. I wish also to congratulate your family that traveled all the way from Italy here to celebrate this day with you. Thank you for supporting this young doctor even today. Now that Francesca will leave again, I'm sure that there will probably be some emptiness feel, but I'm sure that she will continue to bring you happiness on the next steps of her life. I wish also to congratulate your better half for the support throughout this full process and the future milestones.
What a journey since your bachelor degree, Francesca. So many steps forward, and as Carlos rightly said, always uphill, because the simple path was never interesting enough for you. Sometimes the rock were deceptive, like calcium peroxide, tricky and full of challenges. Yet even the most daunting walls need to be climbed, no matter how impossible they may seem at first glance. And you always face them with enthusiasm and determination. Another recurring theme in your journey: trajectories. Strategic and essentials. They guided you to navigate obstacles, finding footholds, and ultimately achieve your goals. This perfectly parallels with your work in biofabrication, refining the 3D bioprinting of bioinks with meticulous precision. Just like choosing the perfect climbing path, finding the optimal printability window requires focus, skill, and a constant drive for improvement. Now, as you stand at the summit of this incredible personal achievement, and what a summit it is, 8C+ grade in your life climb, or your friends at Biotech couldn't be prouder of you. Simone, Francesca, Eugenia, and Lorenzo are cheering for you in the front row, alongside your parents, relatives, and Nico. And don't forget Professor Claudio Migliaresi. Here's to more exciting climbs and even greater success ahead. We can't wait to see you again soon in Biotech, and why not for a climbing challenge at the Infinity Boulder Gym.
Sorry. Let me also take the opportunity to thank Professor Antonella Motta, Professor David Manillo, and Professor Lorenzo Moroni for all the shared supervision efforts and from bringing this dual degree PhD graduation of Francesca to the great results achieved. Coping with one supervisor is not easy, but 4 is yet another big achievement of Francesca. As a final moment, I would like to thank all the members of the committee for taking the time to read critically your thesis and for being with us today. Professor Catherine Levisage, Dr. Annalisa Tirella, Professor João Mano, which could not be here today, Dr. Jules Haring, which could not also be here today, Dr. Samaneh Gaddafi, Dr. Victor Gaspar, which is online, and Professor Martin Honing for adjusting your busy schedule and for joining the assessment committee, and the prorector, Professor Harry Steinberg, for sharing the defense today. I would also like to thank Professor Martin van Griesen for sharing the reading and assessment committee. Thank you for working together with other committee members to ensure this tight schedule that we are always having. Finally, I would like to thank all your friends, colleagues, and scientific family for joining in person, for those of you that could make it, and online. I'm sure that there will be a lot of people online. And to the Bury project partners and collaborators within the consortium for all the support throughout this trajectory. Francesca, Dr. Perin, we finished today with another milestone for both of our lives. I hope we have the opportunity to work together in the future. Hope you make the best of those tools and skill sets that you have mastered along these years. It was always a pleasure to work with you. Thank you.
So, Dr. Perin, I'm going to use officially for the first time your title. How does it feel?
Nice.
Feels nice. Very good. So, Dr. Perin, I would also like to congratulate you on behalf of Maastricht University, of our board of directors, and my congratulation is, of course, the full team. And as far as I understand, it started in 2019, and then you moved in 2022 here to Maastricht. I read your book a little bit, and I was a little bit struck by just 3 words, which shows your emotional art. Yes, do you know what I mean? Have you any idea? Well, I can tell you at the end. At the end, you tell me here, leaving Trento is hard for me. So you really must be really homesick to Trento. To Trento is an important issue. And now I heard that you're moving all the way to Spain, to beautiful San Sebastian. It's a very nice city, I can tell you. Basque country. I think you have to leave your family backwards. So I put in my congratulations also your family who are here. And it is also for you a big drive to come over here, and we appreciate that you took the time and the effort to be with this important celebration. I would like to thank also, which has already been done by Dr. Mota, the members of the Corona. Normally I say that it takes 7 minutes to ask questions, but it takes more than 7 minutes to come over here to ask questions, to read the thesis, etc., and that is much appreciated. I wish you, this is the end of your academic career. It's the best thing you can achieve in the Netherlands here at the academic career. It's the highest, the prestigious what you can achieve. And I would say enjoy it. Enjoy the day. It's today your day, and we're going to celebrate. And by that, I'm going to close, and I'm closing officially with my hammer, and the ceremony is officially closed. How do we proceed? Okay, we're going to have a little reception.